Welcome everyone, or welcome back to the University of Michigan Concussion Center's speaker series. For those of you who don't know me, my name is Andrea Almeida, and I am the Clinical Associate Director of the Center. And I am super excited to have you all here with me today. And I'm even more excited to introduce our guest speaker, Dr. Douglas Smith. Over the years, I've been fortunate to know and have worked with some of the world's leading researchers and clinicians. And the goals of these speaker series are to bring these leaders to you to create a space where you can hear and learn from those that are challenging what we know about concussion and implementing new knowledge into your daily lives. Few housekeeping items before I introduce the speaker. The session is being recorded, as you all saw, and it will be available on our website in a few days. You can access this at concussion.umich.edu. There will also be time after the presentation for discussion and Q&A. So please feel free to enter into the Q&A chat room. Now, I would like to take a few moments to introduce our guest speaker, Dr. Douglas Smith. He is a Robert A. Groff Endowed Professor of Teaching and Research in Neurosurgery. And he is also the Director of the Center of Brain Injury and Repair at the University of Pennsylvania. In addition, Dr. Smith is the Scientific Director of the Big Ten Ivy League Collaboration on Concussion, and he also serves as a member on the scientific advisory boards of the U.S. National Football League and the NCAA DoD Care Consortium, as well as the International Concussion Society. Furthermore, recently, the U.S. State Department asked Dr. Smith to lead an investigation on neurologic deficits induced in members of the Cuban embassy, which led to the discovery of a new disorder known as Havana syndrome. Dr. Smith and his research group have discovered mechanisms of concussion and more severe TBI that lead to progressive neurodegeneration, including CTE or chronic traumatic encephalopathy. His collection of research efforts are represented in over 250 published scientific reports, which have led to many honors and medals, which he told me I'm not allowed to list them all. Please join me today in welcoming our guest speaker, Dr. Douglas Smith.
Thank you, everybody. So what a nice introduction. It's great to be here to see someone who left us. I'm holding against it. But really, this should be informal. Like you don't have to call me doctor, just call me professor. We could take this as a small group. I don't know how it works with people online, but if there's something that's just burning question, you can cut me off and I'll try to answer it for you. So I changed the title a little bit. It doesn't really matter, but it's really about what happens in concussion and really what is the philosophy about concussion. So, I want to start with what concussion isn't, and those people go to the web and they find these diagrams and they say, oh, that's it. And this is what concussion is not, where your brain bounces back and forth like a nut in a shell. That doesn't happen, which is space. Or that you have a contusion or a bleed. That's not concussion. That's a more severe traumatic brain injury. And these kind of very vague ideas are just perpetuated throughout. So let's look at some of the real mechanisms. But before, just think about, we used to think about gray matter versus white matter, and there's kind of debate in the field of which may be more important. But think now about a neural network, a nodal network, where cortical representations and how they're connected play an important role. And think about concussion. Have you seen probably diffusion imaging studies where we're talking about connectivity of the brain is disrupted? And let's look at why that is. And really kind of focus on diffuse axonal injury. And I'm stunned by this quote by W. Simmons in 1803, who really said what we're seeing now about a network dysfunction. We called it a derangement, which is produced in the organization of the brain. So that's just fascinating, because it's really something about the organization in the brain. And why are axons selectively vulnerable? Well, it takes about 100 side by side to be as wide as human hair. And so these are very elongated structures. They have to transport protein huge distances. It's amazing that this is the design that seems to work. And so in the initial phase, what happens when someone has a concussion? And how is that linked possibly with the symptoms? So I want to show you something about a tensile elongation. So the brain is rotated and there's shifting of tissue, and the tissue then can go to tensile, compressive, and other forces. The tensile forces that stretch the axon are quite different than your daily experience. Like if I bend my elbow, I'm extending axons in the nerves pretty far. The excursion could be 70%. And they tolerate it just fine. But if I take silly putty, which is kind of viscoplastic, let's call it viscoelastic, and I pull it slowly, you'll know, just keep stretching. But under dynamic loading conditions, the same material just stretches the same distance, breaks. So it's not how far it was stretched, it was how fast. It's really kind of a classic viscoelastic type of effect. And the end result is this is more what we thought historically was diffuse axonal injury. I'm going to kind of challenge this. This is definitely part of it, where you have transport interruption for a reason you'll see, and then accumulation of transported proteins. And the current hallmark gold standard for looking at this accumulation is the abundant amyloid precursor protein. And you're going to hear the word amyloid again later as well. And this is really a hard thing to model. Like you see lots of rodent models, rats are just party animals like this, but they have a real problem is that you look over to the right or your left, and there's this tiny little brain. It's a 2-gram brain for a rat, and it's tiny for a mouse. And is that the right model? Well, not really, because it's been well known for decades that the human brain is selectively vulnerable, that the mass that occurs, so this is a complicated kind of Greek type thing where you see M's and other types of Greek letters, but basically that's telling us about rotational acceleration is related to the mass effect. So one region of the brain pulling against the other. So that like if you were in a car crash with your dog, this was my dog, and you're both belted and you're out cold and you kind of wake up and the dog's licking you in the face because his little 70-gram brain didn't have the mass to pull against itself like your 1,300-gram brain did, or my 1,700-gram brain did, just mostly CSF. But that's fine. But the idea is that it's really about mass. And so we have to worry about modeling this in various types of, different types of methods. And one way to do that is to go down to the in vitro level where you can stretch axons with the same dynamic loading conditions that you would see in human concussion. So you scale it to human concussion. And in this case, I'm not sure if people can see my pointer, but we at the bottom left, we have micropatterned axon tracks that are spanning populations of neurons. So think of this like a mini brain of white matter axon tracks, and in gray matter, there's neurons. And the axons light up with tau protein, which is enriched, it really belongs in axons as a microtubule stabilizing protein, which will again come up again later. And we can deform just the axon only region by deflecting that one region. And what we saw was quite interesting. If we stretch slowly, they just come back, just like a rubber band, and it looks normal, but under dynamic loading, as in concussion, we saw these undulations. It's like something might have broken and bound them up, and they just never relaxed back to that straight orientation. And we postulated that this was damage to the microtubules in the axons. Microtubules are the stiffest part of the axon. They're also the train tracks, of course, that transport protein. And again, this is a pretty complicated thing with all kinds of Greek letters. But what you really I'd like to look at is that this curly structure is the tau protein. So tau has kind of a resting, a non-uniform type of shape. And think of it like a string of yarn that has a bunch of magnets on it. So in a relaxed state, you relax the yarn and some magnets just get together. And if you cause stretching of the axon, microtubules themselves don't stretch. So think of 2 microtubules next to each other actually have to slide past each other as the axon stretches. As I bend my elbow, this occurs. And the cross-linking, the stabilizing tau protein then has to kind of unfurl and break those H bonds as it gets longer, then they reform as it relaxes back. But we did some modeling and discovered that, under normal conditions, this is exactly right, that you can stretch out, and these red dots would be where these H bonds, the magnets, are being released as you stretch, and they accommodate to this one that's after another, like pop, 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 pop. But under dynamic loading, you're pulling a bunch of these magnets, a bunch of these H bonds at the same time, And this creates huge stress at the microtubule binding domain so that you end up with a rupture that you end up pulling against the microtubules, and that causes rupture. If we look at an electron microscope image, we see that the dark lines are the microtubules here. This is a normal control. And right here, you see these curly pieces of microtubules with protofilaments. They have 13 protofilaments kind of hanging out. You cause immediate damage at the nanoscale. Your brain can literally break at the moment of concussion. And that's a problem because now you derail the transport of cargoes, the proteins, and you end up filling up these axons with protein accumulations. So we see this as a human brain after more severe injuries, since people with concussion don't really typically die. But we can see here's an undulation, and then here's maybe a more evolved area where the transport's interrupted in those regions. And you have those classic kind of swellings of diffuse axonal injury, again here with amyloid precursor protein. But this can't be the whole story, because really in concussion, in an animal model, swine model that we use, we don't see a lot of this APP pathology. And yet we know that in concussion, there's this really overt cognitive dysfunction and other sensory motor dysfunctions. So what else in this field where all the axons had the same mechanical loading? This might be the more extreme extent, but they all got injured. So what's going on with those axons? Well, in the in vitro model, we can look at various types of ion influx, including sodium and calcium. And we see that before and after, at higher levels, here you have an undulation appearing. The brightness tells us that more sodium and calcium came in. And even down lower levels, before you see undulations, sodium and calcium are pouring in. And this is a tough thing, because if you pour in sodium, that's really showing us that there's what's called non-inactivation of sodium channels, that they're not performing right, and too much sodium is coming in. And that means you cannot generate an action potential. The signal generating machinery is taken offline immediately. Like if you were to strangle your neighbor, don't do that. It would take a while for them to lose consciousness. But in concussion, the effect is immediate. Strangulation, that's about blood flow, hypoxia. Concussion is about electricity. You're taking the electricity out of the system right away. And a study that just came out in Active Neuropath, it's not on your coffee table, but it's impact factor of like 18, so we're very pleased. And it kind of this kind of field of normal axons looks kind of like these fish tank, neon tetras. And basically the blue you're looking at is the nodes of Ranvier, and the reds are different kind of connecting proteins. And this is across the white matter showing us the nodal structures. And you might remember that the nodes of Ranvier are really important for saltatory conduction, so you can get 100 meter per second transmission across the brain, which is essential for the timing of the brain. And here's just a more magnified area where we see this intact structure. But after a concussion, we now find that, like again, this is a sham, but in concussion, the green is an APP swelling. But all around it, we see that you've lost either the sodium channels, they've become proteolyzed, we've seen, or there's loss of the attachment proteins, or there's a shifting around, so there's a void in that section, so that you're disrupting the signal generating machinery in vivo immediately after a concussion. And that persists even a long period of time, as you see, at least for 2 weeks. So as you're thinking about people you might see in a concussion clinic, this is what we think their brain might be looking like, because that is possibly the cause for the symptoms, since compared to the swellings we see, that's the majority of the changes in the white matter related to the sodium channels. And if we go to the human, again, you can't get concussion brains from humans, but if we want to see the clinical relevance of this, we see that in human moderate and severe traumatic brain injury, there too we find where APP might be swollen, we see this loss of the sodium channel integrity. And we see these changes in areas you don't see APP changes. And this is important. I'm not going to show you some of the imaging studies we're doing, but with diffusion imaging, there are places that we don't see any changes as far as axonal swellings, but the diffusion is telling us, yeah, there's a lot of changes here in connectivity. We should go back now and look at sodium channels, because what if water follows sodium? And that really explains that change in diffusion. So this is a sodium channel NACH-opathy, or nacho. You end up with nachos in your brain in concussion, where all this, I'm going to spare you the details, but all this very important structure for generating electricity, saltatory conduction is disrupted. So you lose the sodium channels, you untether or lose a lot of this, the support structure. And that's throughout the white matter. I mean, it's really kind of amazing that if somebody's kind of dazed and confused, that's all it is, that you lose this ability to generate those signals that can be transferred across the brain. And we think that is one of the key reasons that can explain that immediate dazed, confused, loss of consciousness, whatever, that it's really a type of nacho that explains this electrical disruption in concussion. And that electricity we find might have an important role in something else downstream. In addition, in parallel studies, we thought, what about if there's a genetic effect as well? And why not at the level of the chromosome? Well, it turns out that I think everybody knows pretty well that if you're running any studies in concussion, males dominate. There's something about the crazy kind of behavior, risk-taking behavior. They dominate. But there are recent data saying that, given the same sport like soccer, maybe females have a higher rate of concussion and a worse outcome, a more prolonged recovery. So could there be a difference if we're finding this with, it's got to be more than this, but could there be a difference in the type of axonal injury that males and females suffer? And looking at back to the in vitro studies, we find that female axons are smaller with relatively fewer and sparser this is a cross-section of axon with relatively fewer microtubules, those dark circles, compared to males. And when there's an injury, there are far more undulations in female axons, meaning more microtubule damage compared to the males. And looking at function, we find that far more calcium and sodium pours into the female axons, given the exact same stretch injury than in males. And what we call synchrony between the nodes, or the kind of a flashing we can see with these fluorescent dyes, is more disrupted in females, where it's not consistently, it's not a consistent flashing that's following a pattern compared to the males that can be restored much more quickly. And then moving on to the swine model, which I was describing briefly before, we see that this is kind of an annotation. The blue dots are showing us where there's APP profiles in the brain. So we have a way to quantify the extent of at least APP profiles. So we'll be working on the sodium channels as well, that the males compared to the females in the same, using the same mechanical loading parameters, which is head rotational acceleration, that we've increased the acceleration because of the size of the brain of the pig is smaller, increased about 7-fold to get the same deformation as in human. It creates concussion. And we can see that there's far more axonal profiles, swollen axonal profiles in the female swine, compared to the male swine. So at least down, again, another thing, down to the nanoscale, there's actually a sex difference. Okay, well, what about recovery? So I just showed you kind of what goes wrong, what might go right. How do you come out of this, massive damage to the nodal networks? Well, think about plasticity, and the term, I don't like the term, it's an accurate term, but people think that plasticity means kind of like some dendritic changes or dendritic spine changes. We know that you can't grow axons across the brain, and that's not going to happen to recover. So this word really should expand to something larger and think of it like that it has something to do with redundancy, which we also have to rethink. And that is that what you learned about the homunculus isn't really quite the whole story. We're all been, inundated with this cut of beef type of approach to the brain and these nodal representations of different parts of the body for sensory and motor function. But there has to be more. So enter the shadow network. So the shadow network sits there all the time, roaring along in the background and listening to every node is listening to every other node. through a system that, is kind of set up, designed by nature, so that you don't just have connections between two nodes. You have maybe a primary job, like this node that in its cortex, let's say, is talking primarily to that node in the thalamus, but it also has axons going all over the brain to contact many other nodes and try to figure out what's going on, just constant surveillance. And I think a really good example of this is like in a stroke, a focal stroke, a person could be aphasic and hemiplegic. 6 months later, and 80 years old, 6 months later, dancing and singing. And then you do functional imaging, you're like, well, wait a minute, that's not what I learned about the homunculus, it's in the wrong place. How could that be possible? You did not grow axons across the brain. Similarly, they don't really do this much anymore, but they would sometimes for epilepsy in kids do a total hemispherectomy. And then we see these kids, later on in rehab, and they're moving all their limbs. So that homunculus would say, this is impossible, and yet they're moving. So there has to be pretty amazing redundancy in the system. And I think it's really just this eavesdropping thing, like, what? That's fine. So while each node has its job to do and take care of the other node and send the information, processing, send on, and that node processing, sends on to the next one, they're all at the same time listening into countless other, maybe all other nodes in this kind of eavesdropping type of thing, this shadow network. And so to examine this, we haven't published this yet, but soon I hope, we take 4 nodes in vitro and we make those axon tracks in between, and we can look at how the node behaves with all the other nodes. And we find that they all go into synchrony, this on and off again type of pattern. So they're all communicating. This is a healthy culture. It's all in unison. And we don't even know how this happens. Like who's, like in the Viking boat, beating the drum, who's telling them they have to pace at this exact level, but they all kind of collaborate and agree that they will go into this synchrony. So here's just normal synchrony. These spikes tell you that the color intensity is going on in a normal fashion. We then injure just one tract between 2 nodes, and the entire system falls offline. So you end up having complete loss of synchrony that may slightly restore over time, but not completely to where it should be. So just injuring one part of this nodal system throws everybody off across the whole brain. In this case, culture. So let's just try to work on a hypothesis about this and how does this recovery occur? And think about under normal conditions, like I said, you have a node here, it has a very firm interaction with another node, likewise over here, but there's eavesdropping going on between nodes. They're just aware of kind of what's going on. And think of it like under normal circumstances, the main job of maybe a lot of you are very familiar with the visual system where photon hits your eye, goes to visual cortex, processing, spins around, processing to all these different nodes. Finally, goes out to the motor system, which then can move the eyes, and you could do like a King-Devic or other eye tracking test, which is really pretty amazing that all this processing is like a baton race. They have to run the right speed, hand it off at the right time, processing, hand it off the next one right time, processing. But in concussion, it's like the baton got dropped. So maybe like conduction velocity and processing speed are similar, that you just can't quite get that signal there on the right time, but the processing is off. Or maybe the signal is completely cut off between the nodes. So the only way to restore that is either revive that connection or have a new way to get to that other node. So right after injury, again, you might have lost the strong signal between the nodes you had. In this case, could be because you just have, let's say, sodium channel changes. You're really not getting the spark of electricity to get there. Up here, you have more severe injuries where you have the axonal swellings and sodium channel changes. So this is really more severely injured area. But the whole thing is really the discombobulation, the dazed and confused brain due to this microcosm of the nodal network being attacked. But over time, maybe if it's just sodium channels, you could pump out all the excess sodium and calcium, restore, maybe put in new sodium channels, and that way you can get this signal back between those nodes. But this damage up here might have been too much. There could be axon disconnection, and we do know that axons disconnect in concussion. We see degenerated proteins from axons in the blood. So that's really permanent brain damage. You're not growing that axon again. But there is a way I think you can have recovery by tapping into that eavesdropping system. It's not immediate, but maybe over time you can condition this to form an intracerebral relay so that the connection between the nodes is now strengthened. So it's, hey, listen, can you do me a favor? Can you just I need to pass on this information. So it's still processed here, sent to the next node, is shifted down to this node that's in the thalamus, and now you have the way to get another way to that spot. And maybe that's what we see with that stroke patient or the total hemispherectomy or in concussion or more severe traumatic brain injury, that you're actually using other portions of the brain. We see that with some of these connectivity mapping, that some of these cortical representations shrink, other ones grow, these connectors sometimes shrink and grow. And this is just a hypothesis, but this is really what might be going on, especially around areas where there's more severe damage. And so, this is, I think, a step towards the future where we will be using these connectivity maps to look at this recovery, which will be really important, I think, in understanding how outcomes differ in concussion and how this person maybe has better improvement than another person. Okay, but what now? And did you ever notice that during the pandemic, I think we were all had a bit of brain fog and had to label things that didn't really need to be labeled. Everybody wonder that? You're not why is it this way? And so, one thing that's pretty obvious is that we know about 80% of patients with concussion recover pretty well. And if you're running a clinical trial, you don't really want to pull them in. They're going to get better no matter what. But for that 20% or so, we know that blood proteins show axon degeneration. That's a way you could enroll them in a clinical study. And it's been 9 years since we had a workshop that identified many, many therapies that could address this. I think that that's on the forefront of the future, maybe the near future, where we're going to look at new therapies, try to actually address something we've diagnosed in concussion. Not that you diagnosed you had a concussion, it's diagnosing the fact that that's underlying concussion symptoms. And another thing we can think about is that mapping that nodal network, that really the superimposed shadow network, because that has to play a lot in recovery. And what if neuromodulation could tap into some of the nodes, stimulate them, just like it's being used for other types of disorders, that you actually enhance communication between these nodes over there that we saw work with various patients. Why don't we try to enhance that in this one patient that we're doing a clinical study? So, maybe this might be a way to address some of the patients. Okay, but then let's turn to the much more chronic phase that you have after traumatic brain injury, whether it's repeated concussion or more moderate and severe traumatic brain injury. And the topic, rather than saying CTE, chronic traumatic encephalopathy, we've come up with a term called TBI-related neurodegeneration or TREND, which I think is more apt, which I'd like to show you why that is. So it's really kind of curious that if you're in the neuro ICU, bless you, that you have people who have about 1/3 of the patients in there develop these amazing plaques, diffuse amyloid beta plaques that look like the same you find in Alzheimer's disease. But they could be really young folks. And this happens within hours of injury. So, this is really remarkable. Where does all that amyloid beta come from? So we discovered in a series of studies that you end up with the APP that we see that we use as a marker of axonal pathology happens to be transported with the same secretases or enzymes that cleavage to the amyloid beta peptide. It's a really sticky peptide. And they're all made within the axon membrane compartment. And then you start building up all these oligomers and fibrils, and then the axon might swell so much that it will actually rupture or pop kind of like a zit and just dump that stuff out into the brain, where it can then aggregate either into plaques or just hang out as protofibrils and fibrils. And here's a plaque in a human brain here, right near axon swellings that are making the A beta. So the axons themselves are the generator of A beta that forms these plaques to an amazing number in an acute setting. But diffuse axonal injury is an injury that keeps on taking. It doesn't stop at the time of injury. We look in the swine model over time, and we see, man, we still find axons degenerating 6 months after injury. And in humans, we find this happens even years, that there's never an end to axon degeneration. And there's never an end to the generation of amyloid beta. So this can be a way to constantly make plaques over the years that may become more fibrillar, more Alzheimer's, more Alzheimer's, mature Alzheimer's plaques. And this is going on at the same time that this is the splenium of the corpus callosum in various patients who have survival. The darkness is inflammation, is a bunch of active microglia. And look over here, this is compared to controls. So you notice the loss of the volume compared to controls, and that's due to this just ongoing loss of axons over time. So the white matter, and I'm sure you've all heard, white matter keeps disappearing in patients with TBI and repeat concussion, but it's not simply just disappearing, it's making weird proteins or peptides on its way to degenerate. And here we can see that later on, what we don't see acutely are more of these fibrillar plaques, the senile plaques, that again, you'd see kind of an Alzheimer's disease system. Okay, but what you've heard most about is tau. I'm sorry for stealing or ruining Shakespeare. So CTE is a really kind of a hot or highly debated topic that's gotten a lot of interest over time. So here's the published publications over time. So you can see around 2010, the publications have gone wild. And there's been a lot of media reports, and I think it's really unfortunate because there are a lot of people really scared out there. Some people who might just have depression will shoot themselves in the heart so they can donate their brains for examination. It's really quite tragic. And many of us get a lot of emails of very concerned people who play contact sports. And the reality is that there really could be something there for some people, but we don't have the epidemiology. And on an individual basis, we definitely can't tell anybody much. I worry that people get PET scans and they find something on them. Well, that's not meaningful at this time. Okay, but what's really kind of been thought to be pathognomonic, and this is really based on 10 highly selected cases that were skewed cases, that the pathognomonic lesion is, you see tau profiles at the depths of sulci. So this is a sulci in the brain, going up to the gyrus. And these dark spots are tau pathologies that include neurofibrillary tangles that are inside the neurons and tau astrogliopathy of these thorny-shaped astrocytes that have this kind of tau accumulation. And this occurs in individuals in both moderate and severe TBI, where we see ongoing axon pathology, but in this case, a dark staining is not APP, that's phosphorylated tau. So this may be one of the generators of these tau pathologies that you see in the chronic state. So that axon degeneration, remember, tau belongs in axons. So axon degeneration can make not only the amyloid beta, but play a role in the tauopathies as well. And here's a single severe, where you've probably heard it's just repetitive, but certainly people with single severe and repetitive concussion have an excess of tangles throughout the brain compared to age-matched controls. I say the repetitives actually seem to have quite a bit more. It's really aging on steroids as far as the total tau pathologies, and especially neurofibrillary tangles. But what doesn't seem to be the case, what we've discovered, so it kind of makes CTE less interesting, is that instead of the pathognomonic lesion of the tauopathy at the depth of sulci, using various stains, we see it's not really neurofibrillary tangles, but just more of them all over, not in this space, and more of them all over, but in the Alzheimer's distribution and in the same phenotypes of tau pathologies you find in Alzheimer's disease. So it's kind of like a additional, moved up in Alzheimer's disease, but not necessarily something unique to trauma. But this depth of sulcite change really could be astrocytes, and what that means has yet to be shown. And there's still debate about this, so we'll see if that comes up. That's just been work we've been doing fairly recently. You can see that I haven't covered everything, but under trend, it's not just depth of sulci, changes of tau. We see atrophy in the brain, inflammation, axon degeneration, amyloid beta pathology. I didn't even get into TTP43 pathology. And all of these changes can happen at various degrees in any individual. So that's why we kind of prefer the word trend, which the NIH actually uses as well. And it may be, and we haven't really found the actual smoking gun for any kind of let's say dementia type of change, but possibly when you start having the clinical symptoms, it's that you had this remote previous history of a single severe TBI or repeated TBI or concussion that accelerate these symptoms. So some epidemiology, at least for a single severe TBI, say that it's an earlier onset of things like dementia, cognitive decline. And repetitive TBI, we don't quite know yet. And how these are related to the neuropathology, such as tau and amyloid beta, we also don't know. Things are very, very early. But the controversy continues. And this is from a few years ago, where people were actually pitting one neuropathologist against the other. And some of us were not very happy with that. So we said, look, you can't just do another shark in the water story, that everybody with CTE, if somebody commits suicide, that's because of CTE, which is kind of I was telling people just shortly ago that I won't mention the person's name, but the person is well known. And so when I looked under the microscope, I know why this guy committed suicide. Like, his microscope is better than mine. That's just craziness. But that's kind of where we are. It's a charged, topic. So to answer this, we formed a Center Without Walls that's funded by the NIH that combines a large group of people who are sharing brains of people with repeated concussions, severe, moderate, severe TBI, neurodegeneration, and controls, so that we can start to really get at what underlies the chronic phase of traumatic brain injury. And then I just want to completely make a segue, a shift that just came out today. We're pretty happy about it. It's kind of a weird little essay on axon stretch growth. So if you want to look at Scientific America today, please. All right. Well, with that, I thank you for your attention, and it's a pleasure. Thank you.
Thank you.
Yeah.
It's a great talk.
Great. Thanks.
So far we have two, well, I guess first, do we have any questions in the audience, in the live audience? And
You said like 80% of patients recover and 20% don't. What is your definition of 20% don't? Like what are their shortcomings?
Yeah. So it's more. Most of the studies have been on cognitive dysfunction, and that's at 3 months. So that by 3 months, they have not, gone back to baseline.
yeah.
Can the audience hear the questions?
I guess maybe if somebody can write in the Q&A that's virtual, can you guys hear us and hear the questions? All right, great, thank you. Eliana, go ahead.
Yeah, so very interesting talk. And I think obviously the kind of story you presented about the axonal injury is really compelling, and the implications for connectivity are big. And obviously I think the kind of natural extension of that is into kind of structural connectivity and white matter connectivity. But obviously we assume that the functional connectome is partially based in physical structural connectivity and partially not necessarily. Do you think there would be kind of similar connectivity-based disruptions at both the structural and functional level, or do you think the functional might be more potentially resilient to, or more able to reorganize to concussive injury?
Yes, I think what we have to do is, and I hope we can help decipher this, is when you see a change in diffusion, and nobody knows what that is. Nobody's even sure that's inside the axons. So I think first we have to decide what that is, uncover what that is, if we want to interpret what those changes mean. And there are a lot of studies say, well, it seems to go up, and it goes, no, it goes, FA goes up or down, or MD, or it's like, and then just looking at a change. So I think we first have to nail down what those changes mean, and then I think we can interpret that. So is it structural? Is it functional? I mean, because you're combining that one thing that's just looking at water protons with another thing that's looking at blood flow, right? And so you're kind of combining two things that are looking at different things. So it's maybe not the right, it might be kind of the square peg and round hole thing, you know, so we have to be sure we're putting that together. And then for the functional imaging, you know, you've probably heard about the, was it the salmon the dead salmon that they yeah, it's all changes. So, yeah, I think we have to go a little further with these types of studies to know, you know, how reliable are they. But yeah, I think that'd be absolutely essential that, you know, not everybody's gonna have depth electrodes, but it'd be really interesting to find out how those changes that we see with connectivity with current technology translates to real function.
I'd never heard of the shadow network before. I think that's kind of an intriguing concept. I don't know how new it is, maybe I'm just not up to speed, but when I heard about it, I thought of like the idea of cognitive reserve. So I guess I wondered, like, do you think that's like a physical or network underpinning of this kind of cognitive reserve?
Yeah, so yeah, I made up the silly term myself, so sorry. Yeah, so I think you're absolutely right. So what if people who are, you know, very active, have very active minds, and read, and like the Nun study about reading, and those who read and those who didn't, those who watch TV, soap operas, and those who read advanced novels. And maybe if you stimulate your brain, you might enhance that network. So that if you start having mini strokes or whatever, you just have another, a better ability to relay signals around the brain. So yeah, that would make sense. I'm guessing here, but I think that absolutely could be part of cognitive reserve, which, like plasticity, is just kind of, oh, cognitive reserve, but that thought bubble for most people is empty, right? What is that? Like a little spare tire someplace? It's not. And the other thing I kind of worry about is that, let's say you do, we're almost certain you form these relays, because, you know, these patients that are losing that representation in their brain, they are obviously relaying that information. So, but there is no spare tire, and that pathway had a job already. Whoever said you use 2% of your brain was using 2% of his, right? Because you're using it all. It's all there for a reason. Evolution is not capricious. And so, you know, what if you kind of burn that area out? And there's some problems, like in post-polio syndrome, people age with, you know, polio symptoms, they actually start to burn out the nerve systems, because it's just like you're overworking. It's like, you know, doing a power drill on a little, you know, extension cord. Is burning out that wire with too much power draw. So there are a lot of things we don't understand about this yet.
I have a daughter who plays soccer and heads the ball. So I'm curious about, have you looked at other sports? Has there been extensive looking at comparing men's and women's brain injuries? And was this just one study? Like I'm not familiar with it, so I don't know.
Yeah, I say that I'm seeing more recently kind of more of a mixed bag. And that's good. I like to have the debate that some people say, yeah, we really don't see a difference. And it could be about reporting. There could be just that the males are less likely to say they had concussion. And I've talked to you know, for Penn football players who admitted having a concussion, but they didn't tell anybody because it was our senior year and they want to finish out the season. And you know, so there could be reporting. There could be a lot of other factors there. But there is something that should be looked at the very least, that there could be a difference. And I think it's just, you know, women's brains on average around anywhere between 8 and 15% smaller than males, but the network looks to be about the same. So it may be that you really have the same, females and males have the same network, but the smaller brain just means that there's a trimming down on size. Or actually, I think the opposite. I think the male brain is bulking up, because that's all about testosterone and a development. And, you know, males have thicker skulls, you know, maybe, and when they look at caveman skulls, I think more than 50% have heel fractures. So this is tough time, you know, back then. And it might just be that this is part of that deal, that males just bulk up various structures in their brain and skull.
And the additional weight of a man's brain wouldn't increase the torque enough.
Well, so it should be worse. Having greater mass should be worse,
Yes,
and yet
yes.
that doesn't seem. It's like the pig studies. The males were slightly bigger brains. We tried to get them even just to make that, but we don't mind that they're a little bit bigger, because since the females had more axonal pathology, it really suggests that's not. It's just excessive to what the males have. That by this thing called a Holborn scaling equation, which looks at the mass of the brain, you're absolutely right. There should have been more damage. Everything else being equal, for the larger brain.
Thank you.
Welcome.
Anybody else?
I'll have an esoteric comment and question. So our
I can't handle esoteric.
Okay, yeah, I figured you wouldn't mind that. So are pig female necks less large than male pig necks?
They're, you know, especially as the age, everything is becomes smaller. The males get much bigger, much faster than the females. These are mini pigs, so they're not like the farm pigs that grow to monsters really fast. But we control the mechanics. So while people have debated about, you know, in soccer, for example, that a more supple female neck might cause more damage, that's not going to be the case here because we have accelerometers where we can precisely control and have the identical type of injury between the males and females. But now that you bring up the neck thing, the one thing that always troubled me is that you're taking, let's say a 200-pound male, two 200-pound males, lunging up through the air and flailing their heads and the two coconuts hit. And you think it's certain forces versus two 120-pound females lunging up and think of the forces that are different. And it could just come from like, I was involved with this, Congress was worried about roller coasters and G-forces. And it's a conceptual framework with G-forces, if you're in a centrifuge, that's a problem, but that's about blood flow not getting into your brain. It's not about your head moving fast or your brain moving fast and having injury. So that's just about, that'd be more hypoxic ischemia. With the head rotational acceleration, it's not so much that you see the thing moving around, it's something your eye can't see. We're talking about 5 milliseconds. So it's really the moment those two coconuts hit. So I think if you're guarded and you're heading the ball, your head doesn't move, your brain doesn't move, you're probably okay. But if you have a head from the side, now that it's not this part when your head's going like that, it's actually that moment of the impact that the momentum of the brain just goes and the whole everything shifts around and causes those damage to the axons. And just one last point: if you have a hammer with a normal neck and a certain weight and a head, and you come down at a certain velocity and hit a nail that you can tell the pressure load, and you get a reading, and you make a floppy rubber neck, and you come down the same velocity, it's going to be less, right? But that's linear acceleration or G forces. What we're really talking about is rotational.
Just like everything else, it's complicated.
You showed one of the later illustrations was from your Connect TBI study and the different types of neurodegeneration, which you literally see upon autopsy when you look at brains. To what extent are you optimistic about the field coming up with biomarkers to be able to measure the presence of these types of neurodegeneration before it comes to the point of doing autopsy? How's the field doing on that?
So I think we have an advantage in a way to neurodegeneration like in Alzheimer's disease. Well, one advantage is we can steal from them. I borrow from them, but the other one is that we know time zero. For Alzheimer's disease, they're getting a bunch of people to volunteer for lumbar puncture that have no idea when, because they don't want to start a clinical trial when somebody is symptomatic because the horse is out of the barn. The pathology has gone too far. But since we know time zero, we know certain people that are at risk. And let's say we do find some link. It can't just be axon changes, but let's say that's part of it, that we do have markers for axon pathology that will be there before you see the tau pathology. I didn't mention we see the amyloid pathology very early, but we don't see tau pathologies till year or years after injury. But if we saw ongoing axon degeneration, then we're like, oh, I think tau's getting messed up, amyloid's maybe being produced. So you could pick those up using like markers of axon degeneration, like neurofilament light, or something we look at a spectrum breakdown product that really seem to indicate that axons are degenerating. And then you could combine that with the up and coming Alzheimer's and other neurodegeneration type of evaluation, like with most of the CSF is for things like amyloid beta, but there are some tau and other things. And now some of the PET scans are promising. So we could just take something that we know time zero, who to look at. So you really narrow the field. That axon pathology could play a role, and then marry that with the current and evolving, neurodegeneration markers.
Okay, so on the chat, we do have why do we believe new axons absolutely cannot or maybe will not grow in the brain?
Yeah, that's a good question. And so you can grow axons if you damage the peripheral nerve. They'll grow about an inch or so a month, which isn't very fast, but they'll grow that fast. And they'll grow because you don't have the inhibitors that are in the brain. So you have something called MEG in the brain. There's another one called no-go, which is literally axon no-go, and it will inhibit axon growth. And it's in high abundance and it's built so that you don't get this crazy outgrowth and everything's connected too much. I mean, maybe I think during development, you could form this shadow network, but you want to keep it to a minimum. And so, yeah, we've never seen anything get beyond a couple of millimeters in the brain. There's a scientist called John Pawluszak, who retired recently, who actually looked at axon regeneration in the brain after trauma in a rodent model. And there seemed to be some kind of new growth cone formed, and then that was it. It just peters out. I think these inhibitors of growth just take over, and they say, nope, it's just all over. And the other thing is that if it were to regrow, how would it even get to the other place? So that last Scientific American thing I was showing was, so stretch growth is there's no guidance of axons. It's actually they've already formed a synapse. They're fully connected. The brain or long bones grow as vertebrae. The axons go along for a mechanical ride. So they either elongate and grow more in length, or they disconnect. So when we're actually looking at morphogenesis of brain white matter, and as the cortical folds are developed and the brain keeps expanding, the white matter tracts have to keep expanding as well. So they don't actually navigate to the direction finally. They did that when the brain was like in an embryonic state. That actual type of growth was not growth cone, it was just stretch growth of connected axons.
Right. Are there any known effects of lifestyle choices, consumption of alcohol, caffeine, THC, et cetera, on the long-term picture for TBI victims?
Yeah, so there's a lot of issues with like I said, alcohol has a very bizarre effect with people, even with concussion, but definitely more severe traumatic brain injury, and even sleep medicine. So something you might have heard about zolpidem for people who have minimally conscious, that this is a sleep medicine that can actually wake people up. So there's a lot of these paradoxical effects. So you have to be really careful and don't treat a TBI patient, let's say with headache, which is so common, with a regular toolbox of headache remedies. You should treat them as a concussion patient, but then individually, and keep an eye and say, okay, we'll try this, but let's see how it works, because they often have very strange responses to what would be thought of as like typical medicines.
And we have, do you suspect the new meds for reducing amyloid plaques in the Alzheimer's patient may be applicable for TBI treatment?
Yeah, absolutely. I mean, unfortunately, you may know that there's been some troubles with those clinical trials. That amyloid might do a really good job of, if you're older and you have leaky blood-brain barrier, you can get plaques inside the blood vessels, and that actually might help stave off the secretion of plasma, constituents, proteins, and whatnot that going out to the brain that could cause inflammation. So some of the earlier trials, there was brain inflammation. They think it's because they eroded this kind of the boy with his finger in the dike got pulled out, and all of a sudden you have this leaking into the brain. But if there's anything that shows efficacy in Alzheimer's, then I think that definitely is something we could consider for TBI. I mean, what if amyloid beta does not kill neurons, but it does affect long-term potentiation and memory. So for all I know, that some of these patients, their kind of cognitive issues could be related to the amyloid floating around the brain and getting into synapses, and that maybe that would be something we want to address. Just get rid of that amyloid, and maybe that kind of post-traumatic amnesia phase, although people don't like that term, but that cognitive dysfunction could be addressed even by reducing amyloid.
And what determines TBI severity, mild, severe, et cetera?
The person deciding what it is. So, yeah, I'm reluctant. Yeah, so I think that we could parse the patients into this group that 80% is going to get better by and large, and the 20% isn't. Of that 20%, a lot do recover in a certain period of time. When I say 20%, it is that 3 months. 3 months is a long time not to get back to school or work. But even then, a lot of them continue to recover while others never recover. But their symptoms are so all over the place they're so different from one to the next. I'd be reluctant to say, mild, moderate, severe, mild TBI. We have to change that nomenclature. And then on top of that, there's something called, people are using as complicated mild, which means your GCS and GOS is like a concussion, or you're at 15 for GCS, but you have a bleed from CT. But we don't think the bleed does anything. So it's just, I think that there may be a new kind of workshop or seminar at the NIH to try to address this. It's not going to be easy, because coming up with nomenclature that people agree on is kind of tough.
I mean, at the 3-month span, what's really playing the role now, right? Is it because they haven't recovered in 3 months, or, now they're depressed and they are isolated, they've missed all the school? Okay. Does axon degeneration generally stay localized to the point of impact of the concussion, or does it spread over time? Can this be combated?
Yeah, what a great question. So it's like, unfortunately, the term diffuse axonal injury is not quite accurate. It's multifocal. And there are hotspots, so depending on the angle of the head rotation, there's different hotspots in different regions, especially at interfaces. So gray-white interface is more at risk, probably because of mismatch of tissue properties there. And so there might be more higher strain in those regions. Around ventricles, the ventricle is filled with CSF, and if an axon is kind of going around a flexure of the ventricle, it can be possibly more injured. If you're an axon track and you go by a vessel, the guys right around that have to go around, they're the ones often we see with the swellings. So anatomically, it's where your position and the angle of rotation, which causes the different pattern of tissue deformation. But I think that it could be addressed in the same way. For all of these, you can address it. So if we use, let's say, a sodium channel blocker, or Taxol, which is used for, let's say, breast cancer, that stops cells from dividing, actually binds very strongly to the microtubule binding site on tau protein. So you lose that stabilization of the ultrastructure of axons. And we find that in vitro, if you put back something like Taxol or another taxane, that you can protect the axon from degeneration. And there's countless other types of approaches. You might help the axonal mitochondria, because they soak up all this calcium and don't really make ATP anymore. So there are many points of attack, but I think it'd be the same really for almost any axon that's interested in the brain.
So I'm going to be mindful of the time. I want to thank you again, Dr. Smith, for joining us and taking the time to come down here, visiting us in Ann Arbor. I want to thank everyone for their time to join us as well. I will be concluding the speaker series, but we will join again in January. We'll send an email out with the date and time and the speaker. Thank you so much.
Thanks, everybody.